everyone, welcome to the next episode of my science podcast. Today I am extremely grateful to welcome Professor Andrew Murray, who is the Herchel Smith Professor of Molecular Genetics and Professor of Molecular and Cellular Biology at Harvard University. He did his Ph.D. with Jack Szostak, constructing artificial chromosomes, and a postdoc with Mark Kirschner, showing that cyclin synthesis and destruction regulates the cell division cycle. So thank you very much for joining me today.
My pleasure.
So we're going to jump straight in. And so may you tell us how you use genetic and physiological perturbations, synthetic biology, and collaborations with theorists to try to understand how cells reproduce, respond to their environment, and evolve?
I suppose the short answer to that question is by mixing them together in a big bowl and stirring vigorously. I guess what I'd say is I think scientists are supposed to have a motto. It's called by any means necessary. It's borrowed from a famous Black activist called Malcolm X, who was once asked how he was going to achieve equality for Black people in the United States in a television interview. And he said, by any means necessary. And the interviewer said, and does that include or not the use of violence? And Malcolm X said, just repeated, by any means necessary. I don't mean that one should do violence either physically or intellectually as a scientist, but I think it's crucial that you try and use all the tools at your disposal to answer biological questions. I see scientists as being engaged in a titanic struggle with Mother Nature, who is trying to keep us from accessing her secrets and is vigorously swatting at us with a very strong hand as we do so. And so I think the secret is to approach problems from a variety of different ways. And one of the things I think that really helps is to make problems seem as simple as they possibly can. And so I'm a huge fan of what I think is the correct use of the word reductionist. It doesn't mean reducing everything to one individual chemical reaction at the time. It means trying to find the simplest possible example of a general problem, studying that while admitting its simplicity, and then seeing if you can extrapolate what you find to bigger and more general problems. So famously, two French scientists, Francois Jacob and Jacques Monod, studied the induction of enzymes, and that led to the discovery of the lac operon and very elegant genetics and eventually molecular biology about how that device for regulating the production of enzymes that E. coli uses to metabolize lactose, how all that machinery worked. And Monod famously at the end of this said, in French, he said, tout ce qui est vrai pour le colibacille est vrai pour l'éléphant. And in English, that means what is true for E. coli is true for the elephant. And you could have a long argument in a bar between scientists who study gene expression in eukaryotes and prokaryotes as to whether it's really true how similar the control of gene expression is in bacteria and elephants. But what you could never disagree with is that the concepts that Jacob and Monod discovered as they investigated gene regulation in E. coli were absolutely essential building blocks to put people in the right direction to study the same problem in eukaryotes. And so we're all about trying to find simple problems and beat them to death.
Absolutely. And so on a similar point, how has the Feynman principle of that what I cannot create, I cannot understand helped you in your research?
So I think it's rather direct that I guess, here's the problem. Scientists lie to themselves all the time about whether they really understand things or not, okay? And painful as it is to demonstrate to yourself that you're lying, it's sort of essential for progress, okay? And so the thing that scientists famously do is to do experiments to test hypotheses, right? And what they will do is they will take a hypothesis, they will make a set of, this is the fabulous scientific method, okay? The scientific method is a bit of a crock, since it starts with a hypothesis and deducing things and making predictions and doing experiments. It sort of ignores where the hypotheses come from in the first place, which is sort of, if you like, inspired dreaming and speculation. And what people tend to do is think of really interesting, exciting hypotheses. They make predictions, they test the predictions, they manage to successfully demonstrate that some of the predictions are correct, and then they believe that their hypothesis is proved, right? Although often this famous thing called Occam's razor, which literally means you should always think of the simplest possible explanation, will admit that there is some other explanation that they actually haven't ruled out, but they decide, like, oh, damn it, my lovely hypothesis is still intact. I'm not sure I want to do an experiment that will distinguish between my lovely hypothesis and this very boring, mundane hypothesis. And so I think the thing that is both an argument for theory, because it forces you to state your assumptions as clearly as possible, and then to ask if there is any possible way that what you claim could be true would really work. And even more so for experimental reconstruction. If you think you know how something works, you ought to be able to build that thing. And if you can't build it, there are one or two possibilities. Either you're a very poor builder, which is often true, or it's not built the way you think it is, right? And so trying, if you can make something and it works more or less the same, if it's at least some pale approximation of the biological reality, okay, it's very important to say this. It doesn't mean that that is how the biological thing works. It is at least how the biological thing could work, because you just made something that mimics it.
Yeah. So in your PhD, how did you go about creating an artificial chromosome? Because that seems like a very large task.
Yeah, and I guess what I would say is it's not quite as big as it might have sounded, okay? And so I was working on a project that I had designed that wasn't really going anywhere very quickly. And my advisor, Jack Szostak, had just cloned the piece of DNA, which is the telomere, that is the end of a eukaryotic linear chromosome. And Jack came by my bench one day and he said, well, we now have in hand all the elements that people think are required to make chromosomes. We have telomeres, we have origins of replication, we have centromeres, which are the structures that bind proteins that build this thing called the kinetochore, which attaches chromosomes to microtubules so they can be segregated from each other when cells divide. And we have genes. We need the genes so we can follow the piece of DNA around, because we can't actually see it inside cells. And he was like, why don't you see if, in addition to the project you thought about, you can just put these things together and see if they work. And so what I'm going to say is, at that stage, people had made circular DNA molecules. And so bacterial chromosomes are circular, but eukaryotic ones are not. And they had shown that if you put a centromere sequence on them, they behaved in a way quite like chromosomes. They were present in a single copy per cell, and the two sister molecules separated from each other at cell division. And so I basically took those molecules and I cut them, and I, in a test tube, attached the telomere DNA to either end, and I put them back in cells. And this is an interesting example of where you should be careful what you wish for. So what you might have wished for in that situation is that that would work, that those small linear DNA molecules would faithfully mimic chromosomes. However, if that was true, your project would be done and you'd have to find something else to do to get your PhD thesis. And so instead, and surprisingly, the molecules didn't behave like chromosomes, okay? And in particular, instead of being present in one copy per cell, they were present in about 10 copies per cell. And instead of faithfully, and the only way to sort of interpret that is that effectively the centromere wasn't working and they were segregating randomly from each other. And if molecules segregate randomly from each other, and you want to make sure that by chance both daughter cells get at least some copies, you should have a lot of copies, because it's like tossing coins. Okay. And so now there was a puzzle. And then there are two aspects of that puzzle. One is what you have to do to make them work better. And the other is why don't they work? And so the answer to the first question is you have to make them longer. Okay. And the answer to the second question is that it turns out the centromeres still work, but for when you have two molecules and you want to pull them apart, to make sure they're both attached to opposite ends of the device that segregates them, the mitotic spindle, they need to be coupled to each other, and there has to be some way of telling that one is attached to one side and one is the other. They actually rely on detecting physical tension. Okay, so you need to hold the sister chromosomes together. And so what we proposed is that whatever held the sister chromosomes together was a topological linkage. Okay, and if you have, and we proposed it that the strands of DNA were literally wound around each other. Okay, so if you have two circles and they're physically interconnected like this, you can't separate them. But if they're two lines, they can just twist around each other. Okay, so that was our proposal. Now it turns out it is a topological linkage, but it's not that sort of topological linkage where the DNA is wound around each other. It's that if my finger going towards the screen is a chromosome, I need two of them, they're two sister chromosomes. There's literally a protein ring that surrounds them. And so later on, Kim Naismith, who's an English scientist, demonstrated that this ring exists. And if we want, we can claim, well, it's still a topological linkage, so we were still half right.
Amazing. That's absolutely fascinating. So my next question is, how did you use the frog egg extracts to examine chromosome segregation in vitro?
So there is a Japanese scientist who worked in Toronto in Canada, Yoshio Mizui. And he started making extracts. I guess let me go backwards. So frog eggs are a millimeter in diameter. They're 1.6 millimeters in diameter. Your typical cell is more like 10 microns in diameter. So for the sake of argument, we're just going to call 1.6 millimeters a millimeter. So it means a frog cell is 100 times the diameter of your cell. But since volumes go as the cube of linear dimensions, a frog egg is 1,000,000 times the volume of one of your cells. Okay, and it's big enough so that you can investigate individual eggs biochemically, and you can use it as a way of testing various factors that drive the cell cycle. Initially, people did this by microinjecting the contents of one egg into another. But what Yoshio Mizui did is showed that you could crush frog eggs. And because they're big, they're sort of fragile. And if you put them in a centrifuge tube and you spin them about 10,000 times the force of gravity, they break. Okay. And when people try and understand what goes on inside cells, if they're biochemists, they have to break cells. When the cells are small, it's sort of hard to break them, and they have to do some violence to them. And they also typically dilute their cytoplasm quite a lot. And if you dilute the cytoplasm, you need to make rather a good guess about the stuff you're diluting it with, because it should have the same ionic composition and pH and so on and so forth as the cytoplasm. If you do it the Mizui way, the eggs are closely packed together. There's not very much dilution, and your guess doesn't need to be nearly as good. Okay, and so he and the students and postdoctoral fellows who worked with him, just kept getting better and better at this. And then while I was still a graduate student, I was helping teach a summer course in Woods Hole in Massachusetts, where there's a famous marine biology lab and has a very famous library. And one afternoon when the students were sort of busy, I went over to the library and I opened a journal called Science. And inside it, there was a paper by Fred Loka and Yoshio Mizui about recapitulating the cell cycle inside Xenopus egg extracts. And I was so thrilled that even though you're not supposed to take the new copies to the journals out of the library, I ran across the street to the lab where I was working with my friend Tim Hunt, who's the person who discovered cyclin. And I showed him this paper. I was like, this is so crazy. You can actually do this, that you can make the cell cycle work in a test tube. So now it's biochemically accessible. And so when I started my postdoc with Mark Kirschner, I souped up a little bit the recipe that Loka and Mizui had invented and showed that you could use it as a very effective way of studying the cell cycle. And eventually, with a graduate student, Carolyn Chamu, we showed that you could actually get chromosome segregation to work. And so now you could study chromosome segregation in a test tube. And the great thing is, once it's in a test tube, you can sort of add and subtract components. Mm-hmm. Geneticists add and subtract components by fiddling with genes. And typically it's much harder for biochemists to do, but by making an extract that will do things in a test tube, you can easily add components because you can just pipette them in and you can subtract them in various clever ways. And so you can start to ask which things are required for particular processes to work.
Definitely. My next question is, may you tell us a bit about your work on the spindle checkpoint?
Sure. So this grew out of the work to try and make artificial chromosomes. So I told you a little while ago that when we tried to first make artificial chromosomes, we made these little titchy, short, linear things, and they didn't work very well. And so one of the ways of following their behavior is to do what's called pedigree analysis. Okay, and in pedigree analysis, you take a yeast cell and you use a needle. It's a glass needle. It's about maybe 10 times the diameter of the yeast cell. But you can pick it up off the surface of an agar plate. And then you actually, rather than moving the needle, you move the plate relative to the needle. You can put it down somewhere else, okay? And so because of that, you can wait until a cell has divided. And once it's divided, you can separate the mother from the daughter and tell who's who, because these cells divide asymmetrically. Their daughters are always smaller than their mothers. And you can pick the daughter up and put her somewhere else. And then you can repeat that process. And so you can build up a pedigree. Okay? And if on a molecule that you're trying to follow, it reproduces, it replicates and segregates as cells divide, you can do all that separation on a medium where cells grow whether they have this molecule or not. You allow them to grow up into colonies of cells, and then you can transfer them to media where they need the gene in order to grow. So now you can tell which cells got your molecule and which, okay? And so you can use that to tell how often you lose the molecule and so on and so forth. And so when we did that for these short linear chromosomes, right? And I'll remind you that we thought what their problem was, it's that the sisters, instead of staying attached to each other, fell apart prematurely. What I noticed, because you have to keep going back and looking at the plates roughly every hour and figuring out which cells are ready to separate and so on and so forth, is that there were cell divisions where instead of the daughter being small and the mother being big, both cells had kept growing until they were as big as each other. And okay, so yeast geneticists call that a dumbbell, okay, because it looks like a dumbbell with a very short bar between the two, right? And that typically means that cells have gotten stuck somewhere late in the cell cycle, and at least some of the time in mitosis, okay? And so the argument that we made is the reason that they're stuck is that the sister chromosomes have separated prematurely. And we knew from very elegant work from an American scientist called Bruce Nicholas that if the two sisters aren't being pulled in opposite directions, the attachment between the kinetochore, the thing that's attaching the chromosomes and the microtubules, is unstable. And so we argued that because these sisters have separated, there's no way to they generate tension because they're being pulled in opposite direction. Once they're separated, you can't generate tension. Okay. And that the cells, quote unquote, knew that that meant that they were not properly attached to the spindle, and therefore activated a control circuit to say, wait, let's see if we can attach these guys properly. So that's where the idea that this checkpoint existed came from.
Right.
And we tried to write up a paper and publish it, and we submitted it, and it got rejected, and we never even resubmitted it. And then I went off and did a postdoc and was busy doing other things. And then I started my own lab and decided, well, okay, the way to really convince oneself that such a machinery exists is to find mutants that destroy it. We had predictions of what their properties were. And so Rin Lee, who was the first graduate student who worked with me and is a very talented scientist, set off to find those mutants. They had the expected properties, and we published a paper saying that there's a spindle checkpoint. And like lots of things in science, we did so simultaneously with another group, because there was another scientist, also an American scientist, Andy Hoyt, who simultaneously had the same idea,
Right.
looked for the same sort of mutants. We discovered that we were doing the same thing, and then we decided we would send our papers off together and hope a journal would publish both of them. And the thing that's truly remarkable is the details of how we looked were different. And so it means that he found 3 genes and we found 3 genes, and none of them were the same gene. Okay. So it was sort of, it was, in some ways, it's an example of why what people sometimes think of as the competitiveness of science, people trying to do the same thing at the same time, is actually often complementation rather than competitiveness, because the details were different, we actually found different things.
Definitely. And there'll be examples of that all over biology. And so that is really important.
And I guess I'll say one other thing, so that scientists are very, very fond when someone else turns out to have done the same thing. And the worst thing to happen is this thing called being scooped. You're working on some problem and you're getting ready to publish a paper, and all of a sudden you pick up your favorite journal and someone has done your project. And so the typical response is to believe that something nefarious has happened. People are like, oh, I should never have talked about our results at that meeting. Those people, they heard and they stole our idea. And it's just completely lunatic, because if someone talked about some project that was well advanced and you hadn't even started, you're really arrogant enough to think that you will start from behind. And so there are all sorts of cases where I know where people have been upset about this, and I know sort of both parties. And they just started working on the same thing independently, but it's really painful for people to admit that the idea I had was not as brilliant and as unique as I thought it was, and someone else thought of the same thing and got there first. And so they're like, they stole my idea.
Yeah, absolutely. So we'll move on slightly now. You've already mentioned that you worked on yeast a little bit, but your group works on experimental evolution using the brewer's yeast S. cerevisiae. Sorry if I pronounced that wrong. Why do you use this organism as opposed to others?
So Saccharomyces cerevisiae, the humble brewer's yeast, we use it, first of all, because it smells good, actually. So if you work with yeast and you open the incubator in the morning where you have these petri dishes. It smells nice. It sort of smells a bit like bread and breweries, whereas if you work with E. coli and you open the incubator in the morning, it has a sort of vaguely sort of fecal smell to it. It's not so great. We work with it because it is the sort of workhorse of eukaryotic cell and molecular biology. It has a bunch of things that make it way the best understood eukaryotic organism. So those include the fact it has a tiny genome, it's unicellular, and you can grow it on all sorts of defined media, that it divides every 90 minutes at its fastest, so you can do things quickly. That there was a positive feedback loop that lots of people who, you know, as you make more progress, people are more interested in working on organisms. And so you build a critical mass, and then things go faster and you attract more people. So it's model organism capitalism, if you like. And then last but not least, and this happens all the time in biology, it turns out that organisms have some property that people have already decided are great to work on, that there was no way that you could know in advance they had, which really potentiates research on them. You know, and people sometimes make jokes about this. So for example, in Drosophila, they have these things called giant chromosomes in the salivary glands, so that you could look at where genes were on chromosomes. Before Thomas Hunt Morgan started working on Drosophila, no one had any idea about this. And then they discovered this. And so people have joked and said that the existence of salivary glands in Drosophila is cytological proof for the existence of God, you know, because it was just so fortunate. And the fortunate thing about the brewer's yeast is that you can direct molecules where to recombine by making double-stranded breaks in them. So when you put in a molecule that has broken ends, they find the same DNA sequence where it exists on the chromosome, and then they recombine with it. So it's super easy. All the things that people get so excited about now with CRISPR, and which you can do in more or less any organism, you've been able to do in yeast since the 1980s by exploiting this fact that by cutting in the right place, you can tell the DNA molecules where to interact. And there was no way at all when people started working with yeast to know that that would be true. It's just luck.
Definitely. My next question is, you found that non-differentiating mutants overtake unicellular populations, but are outcompeted by multicellular somatic cell-producing strains. How did you then conclude that multicellularity confers evolutionary stability to somatic differentiation?
Okay, so that's a long and somewhat complicated question. And so the question that we set out to ask, and this is one of these Feynman, what I cannot create, I cannot understand things, was to ask about the two things that make fancy multicellular organisms so exciting to people. Okay, so one is that they're made of more than one cell, and the other is that the cells do different things and they interact with each other in complicated ways. So you have neurons that transmit signals to each other, you have photoreceptors that detect light, you have taste receptors that detect taste, you have red blood cells. It's all organized in this supremely complicated community of interacting cells, and the cells have different properties. And so, but there are also things which are just collection of cells that all look like they're doing very much the same thing. So they're multicellular, but the different cells don't do different things. So there's an interesting question, which of those things came first? Right? Because in principle, you could have groups of related cells that were floating around separately that did different things, but they cooperated with each other and they would be in one big genetic lineage. And But it looks like the order of events has been that you make collections of cells that are all the same, and then you evolve some of them doing different things. But like everything in evolution, looks like is the key word. So since you're in England, you know all about the TARDIS and Doctor Who, right? So the thing, there's a real problem with studying evolution is the fact that the TARDIS is missing, that we can't contact Doctor Who and get aboard and go back and harvest samples. So instead of making inferences, we know what actually happened. And so our approach is, well, can't find TARDIS. And so we might as well do experiments in the lab, either by trying to evolve organisms in the lab or by trying to reconstruct things that could have happened and see if they work. And so Mary Wall, who was a graduate student who worked with me, decided that let's see if we can make two sorts of systems. One is a system where there's a germline and a soma, but the germline and the soma are separate cells, individual cells. Mm. And the other is they're multicellular aggregates. And so in one aggregate, you can have germline and soma. Okay, and so we used a trick involving inducible recombination that would convert a germline, which reproduced slightly faster, to descendant cells that were a soma, which would perform some public good that both they, the soma, and the germline depended on. Okay. And now you can ask, can you do that successfully? Can you do it as multicellular aggregates? And can you do it as single cells? So the answer is yes. And then you can ask, well, okay, people worry an enormous amount about cheating in evolution, right? There are certain things in biology that people, I think, because of their anthropomorphic connections, obsess perhaps a little bit too much on. One of them is cheating. Another one's aging. Another one is, why are we different from chimpanzees? Yeah. Okay? And because we're human, these things seem especially interesting to us, and especially we all believe that other people cheat and we don't, because we're not very good at examining our own behavior with the same critical lens we use on other people. And so we ask the question, now we have a unicellular population, germline and soma, and we have a multicellular population, germline and soma. What happens if we seed them with cheats? Okay? So the cheats are cells, germ cells that never give rise to soma, right? So I told you that the soma divides slightly more slowly. Mm-hmm. So the lineage will propagate faster if you never give rise to somatic cells. The somatic cells, however, produce the public good on which everyone depends. But if you're a cheat who never gives rise to somatic cells, and you're in a test tube with a population of cells that have both germline and soma, you can exploit the public goods that the soma of the other lineage makes. And now you will eventually have a horrible tragedy of the commons, because you'll proliferate faster than they will, right? So you'll drive their frequency down. Yeah. And everything will go badly wrong, okay? Whereas if you have clumps of cells, the public good, which in this case is glucose and fructose, it has to move around by diffusion, okay? Now, even if you're shaking the test tube, around each clump of cells, there is an unstirred layer of fluid. Mm-hmm. So the concentration of the public good is going to be higher there than it is out where the cheats are. Yeah. And so if the cost of making the soma is smaller than the advantage that you get from being close to the public good, you can fight off the cheats. Okay? And that turns out to be what happens in the experiment. Okay? So that's a construction experiment, right? So the argument is, because that strategy is not susceptible to cheating, it's more like, let me put it this way. If that's how you evolve multicellularity, you don't really have to worry about cheating. So you only have to do one thing. You have to do two things. You have to evolve a way of cells sticking to each other, and you have to evolve a way that they differentiate.
Yeah.
You don't have to, get people to show you I'm not a cheat passports. Okay? Whereas if you do it the other way, you're vulnerable to cheats. Okay, so if you evolve differentiation first, you now have to evolve the ability to stick together before the cheats do you in.
Yeah.
Our argument is the second way is more complicated than the first, and therefore the first probably happened.
Yeah.
Okay?
That's absolutely fascinating, and it's a big line of biology, so it's a huge discovery. So back to the yeast. Have you found that when yeast cells respond to a sudden starvation, the arrest destabilizes the genome? And then how can the cells then adapt to start dividing again afterwards?
So what I will say is, so this is sort of a confession, and maybe the people at NIH will hear your podcast and maybe they won't. So what scientists do, and they do it in every country, is they appeal to funding bodies for money to do their research. And so you tell them all the great things you're going to do.
Yeah.
And NIH stands for the National Institutes of Health. So they're the people who support most what's called biomedical research in the United States, which really means cellular, developmental, neurobiological, molecular biology. And so you always try and convince them that your work will ultimately be applicable to public health. Okay. And as an example, the artificial chromosomes that I made as a PhD were modified by other people to be used as cloning vectors. Increase the rate of mutation or caused forms of genetic instability. We've tested, as far as we can see, it does not.
I think you cut out slightly then.
Yeah, no, it just said the internet connection was unstable.
Okay. Okay.
Now, do you want to try and ask the question again, or should we just let it go?
We can move on because we've got quite a lot.
Yes, I see that there are quite a lot.
Okay. So my next question is, the kinetic properties of the conjugates of the N-terminus of cyclin fused to a foreign protein indicated that cyclin is degraded by ubiquitin-dependent proteolysis. What further conclusions could you make from this?
I guess what I can say is the original experiments that showed that cyclin destruction is mediated, is the result of ubiquitin-dependent proteolysis, didn't have all that much to do with me, and my name was generously put on the paper by kind people. And the person who did the experiments, Michael Glotzer, he denies this fervently, but it's true, that he discovered this in the sort of most fortunate of accidents. So he was making and following the destruction of radioactive proteins. And so in the old days, you followed their behavior by running them on gels, which separate proteins by their molecular weight, and exposing those gels to X-ray film. And then the radiation would expose the grains of silver in the film, and you develop it actually with exactly the same sort of machine that doctors used to use to develop X-rays. They were called exomats. And research labs all had dark rooms with exomats in them. And so anyway, so Michael did an experiment where he used a radio-labeled derivative of the N-terminus of cyclin. And he ran a gel, and then he went on a skiing vacation, and he forgot to develop the gel before he left. So he went off and skied for a few days, and he came back, and then he developed the piece of X-ray film. So in the place where the radio-labeled cyclin construct was, there was the blackest of black bands, hopelessly overexposed, right? bad graduate student, shouldn't have gone on ski trip. But then what you could see very faintly above it is there's a little band here and a little band here and a little band here and a little band here. And they're all separated by roughly the same molecular weight, as if you added 1, 2, 3, 4, 5 of something. And that molecular weight turns out to be identical to the molecular weight of ubiquitin, which is a small 76 amino acid protein. So that was what gave rise to the idea that cyclin was ubiquitinated. And so in Mark's lab and independently in Kim Naismith's lab, and there was one other lab, and I can't remember which one it was, people eventually went off to discover this thing called the anaphase promoting complex, which is the device which puts the ubiquitin on the cyclin and sends it to the cell's equivalent of the garbage disposal in the parental sink, where they're, instead of being whirling blades, they're proteases that chop proteins into small peptides. So that's how cyclin is destroyed, okay? So that told people how cyclin is destroyed. And then when I was a postdoc, I had shown that if you block cyclin destruction, you can still separate the sister chromatids, okay? So you stick the cell in mitosis, but instead of being in metaphase where the sisters are stuck together, now it's in anaphase where they've been pulled apart. And so we then exploited that to show that there was actually some other protein that had to be degraded by the same machinery, okay? And a guy called Jack Szostak discovered what that protein is. It's a little tiny protein called securin. And remember, there's this ring that holds the sisters together?
Yeah.
The ring is cut by a protease called separase.
Okay.
And so if you think of separase as protease trying to chop things, securin is just a small molecule that gets stuck in its mouth. Okay? So now you can't chop the cohesin and you can't separate the sisters. But the anaphase promoting complex tags the securin with ubiquitin, which sends it to the garbage disposal.
Okay.
And now the chopping is active.
Okay. That's fascinating. And all this links into an accident in some way. So it's really, because there's lots of examples of that throughout the history of science. And
absolutely. Yes. So if you ask any scientist who's been successful, even scientists, most scientists do have, reasonably large egos, how often they've been good and how often they've been lucky. They will confess that many of the times they have been lucky rather than good.
So we'll skip a few questions now, but if you like, I can link them next to the podcast if anyone wants to do any further research and answer the question themselves. So what did you find when testing whether ploidy, or the ability to perform homologous recombination, influenced the evolutionary fate of budding yeast as it adapts to constitutive DNA replication stress?
I think the main thing we're finding so far is it's difficult to publish papers. So the paper is on the archive, and it's now been seen by various journals who decided it wasn't interesting enough for them to publish. So poor old Marco Fumasoni, who's the postdoctoral fellow who did the work, and I are struggling to get the paper published. So what I will say is, we did these experiments because we're interested in what people call evolutionary trajectories, the sequence of mutations that improves the fitness of cells. Okay. And so Marco set out to do an experiment to ask how cells dealt with constitutive replication stress, which was caused by removing a particular protein. And he did that in haploid cells. We're able to do recombination because it's easy to do it that way. But he also did it in parallel with diploid cells. And so, for example, the reason to do it with diploid cells is when we do experimental evolution in the laboratory, the vast majority of mutations that help our cells grow better are not mutations that make proteins work better, but mutations that make proteins work worse. Okay. So, and I guess one way of thinking about this is imagine your car had lost power and you wanted to accelerate it as fast as possible.
Mm-hmm.
You don't know very much about the internal combustion engine. One way to make your car go faster would be to take all the seats except the driver's seat and take them out, and take out all the insulation, take out the spare tire, do everything you can to lighten the car.
Yeah.
And a famous English racing car designer, Colin Chapman, his motto was just add lightness, right? And now everything works better, accelerates faster, corners better. And so cells, at least in the laboratory, appear to be the same. It's hard to make things work better, but it's easy to throw out the seats.
Okay.
Right? And so the interesting thing is, in a haploid, to toss out a seed in this metaphor takes one mutation. You just have to inactivate a gene.
Mm-hmm.
Okay. In a diploid, you typically have to make two mutations because you have to inactivate both copies of a gene.
Right.
Right? And so now it should be harder to improve things by making recessive loss of function mutations. And so one of the reasons for doing the experiments was to ask, do you have mutations in the same genes if you do it in a diploid and a haploid? And the answer is yes and no, because sometimes you do actually mutate both copies of the gene in a diploid. But the main take home from those experiments is, although as you manipulate what we call genome architecture, whether they're haploid, whether they're diploid, whether they can do recombination or not, you change in detail which genes get mutated. You don't change the processes they get mutated in. So one of the ways biologists talk about how cells work is that they have what are called functional modules, things that do particular things like replicate the DNA, hold the sister chromatids together, make a checkpoint for damaged DNA. It's sort of like in whatever word processing program you use, there's a spell checking module.
Yeah.
And some group of software engineers went off to a corner and worked on the spell checking module, and then some design team integrated that into the rest of things. And so what Marco found is, even as we change the genome architecture, which changes in interesting ways the types of mutations we get, we still hit the same modules.
Right.
And we're still trying to convince scientific journals that that's interesting enough for them to publish.
Well, I think it's definitely interesting. And because DNA replication stress is a condition that it characterizes a lot of cancer cells, doesn't it? So I'm sure that could be an area that could be gone down, and it could be more interesting to journals if it would make it so that it would publish.
We have made that argument in the paper. Yes, and I guess what I would say is that, for the people who listen to your podcast, this is an interesting example of how you're supposed to talk about what you do, right? So I guess our belief is you're supposed to point out interesting connections, but you're not supposed to hype them.
No.
Right? So when you send a paper to a journal, you write a cover letter telling them how great your paper is. And so in the cover letter, we say, these processes probably go on in cancer, but because this is a paper about yeast, we mention that as in the discussion, the sort of inference and speculations, because we're not claiming that we've discovered anything about cancer. You could instead put the word cancer in the title, right? And maybe that's a better way of getting your paper published, even though your paper isn't actually about cancer.
Yeah. So my final sort of area that we're going to go down is, what are circadian oscillators, and how do these relate to your research?
So, Greg Wildenberg, who was a postdoc in the lab, evolved in the laboratory, our friend, the brewer's yeast, to have a circadian oscillator, okay? It's not a circadian clock, which you have, because you can't easily reset it, right? Like if you flew to San Francisco there would be an 8-hour time difference, and you'd be confused for a couple of days because your body would be telling you it's a different time from your wristwatch or your cell phone. Okay, but your body would eventually adjust. So we just built the oscillator that lies at the bottom, but not the thing that will adjust things. And we did that at some level because it relates to people's debates about evolution. Okay, so there is a debate between people who believe in evolution and people who believe in something called intelligent design, which means not evolution. And their argument ultimately comes from an 18th century British clergyman, Paley. Okay, so he wrote a book about the intelligent designer who was God. Okay, and his argument was that if you walked across a heath and you tripped on something and fell over, and since you were a clergyman, you would not swear, and you looked and what you tripped over was a stone, you'd just be like, oh, damn stone and damn me for being so careless. But had you tripped instead on a pocket watch, you would have picked it up and marveled. Because if you looked at the front, you would see the hands and the elaborately painted numbers. And then you'd open the back and you'd see all those wheels and gizmos whirring about. And you would be, this is remarkable. Some intelligent being must have designed this. And Paley's argument is if you open up an organism with a knife, it looks similarly wonderful and spectacular. And so someone must have designed that and they must live upstairs. Okay. And the intelligent design argument is that there are certain things which have a property which they refer to as irreducible complexity, that there's no way you could evolve this in the way that Darwin and Wallace proposed, that you require changes step by step. Couldn't happen. And since the fact that Paley's argument was about a watch, that's why Dawkins' book is called The Blind Watchmaker, because that's what evolution is. And so we decided it would be fun to see if the blind watchmaker will work inside a laboratory, because that means either that the intelligent design proponents are wrong, because we could evolve a circadian oscillator, the thing that lies at the heart. It's just a watch without being able to adjust it, right? You know, either that argument's wrong, or God is testing my faith in evolution by reaching down his or her finger and stirring it around in my experiments. And, you know, I believe in assuming relative probabilities. I don't think that's happening. So that's why we did that experiment.
Okay.
And what's interesting is, and we identified the mutations that are responsible and we put the mutations back together. You know, this is part of the, if I can reconstruct it. And it really works as a circadian oscillator.
Yeah.
We even begin to understand how those particular mutations produce this oscillation. No, we do not. So that's a subject for future research.
Definitely. And that brings me on nicely to my final question, is that what are your hopes for the future and what would you like to research into next?
So I guess my biggest hope for the future is that we as humanity sort of sort ourselves out and don't destroy ourselves and our planet. In terms of research, what I would like is to see us really increase our understanding of how cells work. So it's my belief that if we really genuinely, truly understood how cells work, all the other things about how cells interact with each other would be easy to sort out. And so that what we should be doing is trying to understand how cells work. And I guess one way of saying it is, if you were starting as a graduate student now, what would you work on? I would work on these remarkable things that live in, they're bacteria that live inside insect cells. They've been living there for 150 million years. And there are two classes. There are ones that still have sort of all the genes that you need to keep a cell alive. but they've really been reduced down to pretty much the minimal set. And then there's another class where they clearly have gone further. And so they're on their way to becoming organelles. So we can actually sort of rewatch the domestication of one cell inside another. And there's one sort of crazy project that we're trying to do on that. So a very bright and ambitious graduate student, his name is Tanush Jagdish, he's Indian. He wants to recreate the original endosymbiosis that gave rise to eukaryotes. So he is going to try and persuade an archaean to swallow a bacterium and recreate the original endosymbiosis. And so we're going to give that a whirl, and then we'll either succeed or fail, and then sort of my time will be up and I'll retire and leave it to the next generation of scientists.
Well, that's absolutely fascinating, and I hope that it works. That's all my questions, and so unless you have anything else that you want to talk about.
No, not at all. Thank you so much. It's really been a pleasure talking to you.
Thank you so much for being here, and thank you everyone for listening.